Evening viewer, welcome to our Globe to viewers most amazing news for viewer. Yeah, thousands of tiny changes in the DNA sequence of the human genome have been linked to an increased risk of cancer. Yeah, until now it has been unclear which of these changes directly contribute to the uncontrolled cell growth that defines the disease and which are simply coincidences or minor flares. Stanford researchers conducted the first large-scale analysis of these inherited genetic changes known as single nucleotide variants. Yeah, their study identified fewer than 400 variants that play a key role in triggering and sustaining cancer growth. These variants influence several critical biological pathways, including those that control DNA repair, energy production, and how cells interact with their microenvironment. By highlighting these common genetic factors, the findings could open the door to new strategies for preventing or slowing cancer growth. The researchers also believe this knowledge could improve genetic screening, helping to better assess an individual's lifetime risks of developing cancer. We distilled a large compendia of information from millions of people diagnosed with any of the 13 most common cancer types, which constitute over 90% of all human malignancies, said Paul Khavari, MD, PhD, Chair of Dermatology. This enormous funnel of data allowed us to identify 380 variants that control the expression of one or more cancer-associated genes. Certain variants, if you are unlucky enough to inherit them from your parents, can increase your risk of developing many types of cancer. Kaveri, who is the Carl J. Harjok Professor in Dermatology in the School of Medicine, is the senior author of the research, which was published 17 February in Nature Genetics. Former graduate student Laura Kalman, PhD, is the lead author of the study. The risks we inherit. The study focused on DNA sequences inherited at conception, known as a person's germline genome, rather than mutations that can accumulate during a person's lifetime as cells divide during development or to repair injury. Examples of all known inherited cancer-associated mutations are the BRCA1 and BRCA2 genes that confer a significantly increased risk of breast and ovarian cancers. However, only a few of these high-profile mutations are currently used to protect and also the predict cancer risks. Bear the exclusive report.